Welcome to today's tutorial. We're going to be talking about how your body fights diseases. In other words, you can call it the immune battle. Every second, your body is fighting invisible enemies, the bacteria, the viruses, the toxins, and harmful substances. But how does the body recognize these dangerous invaders? The answer lies in one of the most fascinating biological reactions ever discovered: antigen-antibody reaction. So in this tutorial, we are going to explore how the immune system identifies threats, produces antibodies, and destroy harmful organisms with antigen, incredible as you can precision. See, is any substance that do what? That invade into your body. When you are asked what antigen is all about, you can say that antigen is a substance capable of triggering an immune response. Antigen are usually foreign substances such as the bacteria, the viruses, the fungi, the toxins, or even pollen. They contain special molecular marker called epitopes that the immune system recognizes as a dangerous stuff. Antigen can be foreign proteins or molecules, and epitopes are the recognized portion. What is then antibody? You can say that antibodies are specialized proteins produced by B lymphocytes, also called plasma cells. Their job is to recognize and bind specifically to antigens. It is important to note that each antibody is uniquely designed to fit a specific antigen, just like a lock and key. So note, antibodies are immunoglobulin produced by B plasma cells, highly specific to antigens. There are 5 major classes of antibodies: the IgG, IgM, IgA, IgE, and IgD. And each of them performs a long-term unique function. IgG provide long-term protection, while IgA protect body surfaces. IgE involved in allergies, and IgD helps activate immune cells. The antigen-antibody reaction is extremely specific, and that is explained by the lock and key theory. The antigen acts as the lock, while the antibody acts as the key. Only the correct antibody can bind to its matching antigen. It is important to note here the specificity of the reaction, the lock and key relationship, basis of immune recognition. You can see the creative biological lock and key concepts here. Then let's look at the primary immune response. When an antigen enters the body for the first time, what the immune system does is that it takes time to recognize it. This is called the primary immune response. So during this stage, IgM antibodies are produced first, followed later by the IgG antibodies. So immune cells dictating a new invading virus, as you can see here. Then let's go to the secondary immune response. When the same antigen enters the body again, the memory cells quickly recognizes it. The immune response becomes faster, stronger, and more effective. And this is why vaccines work. So you can see the powerful immune memory response showing rapid antibody production after the second exposure to a virus. Faster antibody production. So this is an agglutination reaction stage. Watch and see what is being explained in this video.
In this blood typing test, we observed the reaction with anti-A antibodies. The antibodies bind to A antigens on the red blood cells, causing them to clump together. This agglutination indicates a positive result for the A blood type.
Do you understand what that was explained there? That agglutination reaction just helped the immune system remove pathogens and is widely used in blood grouping tests. Okay, now we are in precipitation reactions. In precipitation reactions, the soluble antigen combine with antibodies to form visible insoluble complexes called precipitates. So it is important to know that soluble antigen is involved, formation of precipitate, used in laboratory diagnosis. As you can see here, this is microtube. Now we are in complement fixation. Sometimes antibodies activate protein called complements. These proteins help destroy pathogens by damaging their cells' membranes and enhancing inflammation. So note pathogen destruction. Antibodies can neutralize toxins and viruses by blocking their harmful effects. So this prevents the pathogen from entering or damaging your body cells. Very important. Block harmful effects, stop viral entry, and all that. Opsonization occurs when antibodies coat pathogens, making them easier for phagocytic cells to recognize and destroy. You see it here. It enhances phagocytosis, antibodies coat pathogen. This is what happened in opsonization time. So you can see the doctors all over the country, the US, the UK, Nigeria, they are using antigen antibody reactions in medical laboratories for their findings during COVID-19, the malaria time, the hepatitis, the HIV, and all that. Their findings, they introduce drug vaccines that are harmless antigens into the body to stimulate antibody production and memory cell formation without causing severe diseases. That's the research that yields or give birth to the drugs we use today. So in conclusion, my students, antigen antibody reactions are the foundation of immune protection through specificity, memory, and coordinated defense mechanism. The immune system protects us from countless diseases every day. So understanding this reaction helps us appreciate vaccination and the incredible intelligence of human body.